Hello guys, today we are going to discuss a few slides on human chromosomal inheritance, where this human chromosomal inheritance is based on Mendel inheritance patterns. This is to study certain genetic disorders which are following the Mendel inheritance patterns, where these genetic disorders is with human being. So here we have given the caption to understand how conditions are passed down through generations. An accurate family health history is a valuable tool to illustrate. Here within this particular lines or the phrases, we will come across two different things which we have to understand. One is that to understand the inheritance pattern of the disease, and also the complete family generation of that particular proband or the individual with whom we have identified the genetic disorder. So here within this these slides we are going to discuss the autosomal related and sex-linked related kind of the genetic disorders which are dominant in inheritance pattern and recessive in inheritance pattern. So we are going to get into the first slide. The first slide shows a complete brief flow chart like formation which I have prepared with some consciousness and making the information into a concise form, where this will is going to be act as the introduction to the whole slides which we are going to discuss further. So here we can see the DNA, chromosomes and the genes. Here DNA is one of the expressive character. Whatever the part of the DNA is expressive, that is the expressive character that is only considered for the study, and this DNA is going to get condensed into the chromosomes. So here the same DNA is going to form the unit of inheritance of character, which is called to be as the genes. It is going to present as the form of the alleles, which is the genes alternative form, that is the two forms of the genes, while it is going to express in the form of the trait. So these alleles are going to express as dominant and recessive trait. Now here the DNA and the form of the genes is going to present on the chromosomes. Now here you can see two arrow marks which are indicating towards the chromosomes has been shown. Here chromosome has linear expressive loci. Each loci is a gene which expresses the character. Now again, these chromosomes having the gene in the form of the alleles, any of the alleles which is the dominant or the recessive form, is going to present either on the autosomes or on the allosomes. So here, because of the presence of these genes in the form of the alleles, that is the dominant expressive trait or the recessive expressive trait, if they are going to present on the autosomes, it is autosomally dominant form of the inheritance. If it is the recessive form on the autosome, that will be the autosomally recessive form of the inheritance. If it is present on the allosome, here if the dominance 1 is present on the allosome, that is the allosomally form of the dominant. But here we are not going to call it to be as the allosomally, because in the human beings there are only two allosomes, that is X and Y. XX is the one and XY is the other. XX is for the XX is for females and XY is for the male. Y doesn't have many characters or the genes to which are going to inherit the many number of the whatever the uh, somatic parts or the somatic whatever the expressive characters compared to the X chromosome. So here we are going to consider only the X chromosome in the at the place of the allosomes, and that is going to comprise to form the dominant. So here X-linked dominant trait is going to be considered and X-linked recessive trait is going to be considered. For Y chromosome, we are going to call only it as the Y-linked genes. Okay, so here Y-linked genes is the different. We are going to create a separate slide for this Y-linked genes, but in these slides we are going to create only the autosomally related dominant and recessive and allosomally related X-linked dominant trait and X-linked recessive trait. I hope this slide has been clear and I had been audible for you. And further, we are going to move to the next slide. Human chromosomal inheritance. Inheritance of traits for humans are based upon Gregor Mendel's model of inheritance. The traits are inherited by genetic material in the form of the chromosomes. Mendel has proved that the inheritance of traits depends upon discrete units of inheritance called factors or genes. Factors in nothing but which are going to influence to express the character. Here the same thing is going to be done by the genes. That is what Mendel called it to be as the factor or genes, but we are going to consider genes here majorly. The Human Genome Project estimated that humans have between 20,000 and 25,000 genes. These 20,000 and also 25,000 genes comprises of qualitative kind of the inheritance, that is the biallelic form, that is the dominant and recessive, multiple allelic form of the inheritance, and quantitative type of the inheritance. So here these all three characterized groups has been included in this 20,000 to 25,000 number of genes. The different forms of the genes that are found at a specific point along a given chromosome are known as alleles. Different forms. Here, for example, as we had discussed that in Gregor Mendel, if you are going to take in pea plant, the seed structure round or wrinkled, the seed color yellow or green, or the pod shape, or if you are going to consider in the human beings, the hairs of the human beings curly or straight, or eye color brown or black. So in this, one is going to be the dominant and other is going to be the recessive. That is because of the two different natures of the same gene to get expressed. Those two different natures of the gene, either of dominant or recessive, both, is called to be as the alleles. In a population, a number of alleles are found for a given gene. So here in a population, there will be number of alleles are found for a given gene. For example, if you are going to consider in a population straight or curly hair, there will be the hair structure that is straight or the curly. If you are going to consider within the population, there will be two alleles of the same gene, but they are going to express a little different from the other individual, but the character of the expression of that particular allelic character is going to be the same, but due to some external factors influence, there will be the difference. That is what the explanation here. But the gene being to be having a qualitative kind of inheritance, that is going to have dominant and recessive kind of the expressions. Diploid organisms have two alleles for each autosomal gene, one inherited from the mother, one inherited from the father. Diploid organisms in the sense they are going to have two sets of the chromosomes as per their total chromosomes. One set is going to come from the parental side, that is the father side, and one set is going to come from the mother side, that is the paternal and maternal. Human beings have 46 chromosomes, 22 pairs of autosomes, and one pair of sex chromosomes X and Y. Now here, as we have spoken this previous slide, that diploid organisms have two alleles for each autosomal gene, one inherited from the mother, one inherited from the father. Here, if you are going to consider this 22 pairs, those are going to come from this 46 total number of the chromosomes. Out of this, X and Y are separate, those are the allosomes. The rest 44 chromosomes comprises to 22 pairs of the autosomes. So each pair of the autosome is going to have their own number of the genes which are linearly arranged on them. So here, when they are linearly arranged on them, these homologous pairs, they may have the allelic form either in the form of the homozygous type or heterozygous type. Homozygous type can be of dominant or recessive. Heterozygous type, you know that it is going to have one dominant and the other recessive kind of the allele of the same gene. Now, next point. Individuals with two copies of the same allele are referred to as homozygous for that allele. Individuals that have copies of different alleles are known as heterozygous for that allele. Already I have spoken about this. The inheritance patterns observed will depend on the allele is found on either an autosomal chromosome or a sex chromosome, and the allele is dominant or recessive. So here we have already spoken that within the inheritance pattern or with the help of the inheritance pattern, we can understand or observe that the allele, either it is present on the autosome or on the sex chromosome. So with the help of those pattern of inheritance of these autosomes and sex chromosomes, we can understand that this gene is present either on the autosomes or on the sex chromosomes, or whether it is a dominant one or whether it is a recessive one. This thing is we are going to understand. So modes of inheritance for single gene is autosomal dominant, autosomal recessive, X-linked dominant, and X-linked recessive. So these are the modes of the inheritance. So here a dominant, a gene which is dominant in its expression, if it is present on the autosome, it is going to show the different type of the inheritance pattern compared to the rest of these three. In the same manner, if it is autosomal recessive, it is going to show the different pattern of the inheritance compared to the rest of these three, that is autosomal dominant, X-linked, X-linked recessive. In the same manner, this is X-linked dominant is going to be very unique in its inheritance pattern compared to the others, and X-linked recessive also will be very unique in its inheritance pattern compared to the others form of the inheritance within this group from this autosomal to the last. So here next is we are going to discuss these slides, that is a particular brief description or describing of the autosomal dominant inheritance pattern. We have taken the hallmarks in the sense what particular is very important to understand within this dominant inheritance within the autosomal form. Those points which has been taken here, and we are going to discuss on that. Now here the expression of dominant gene is sufficient if only located on one of the autosomes, can cause a particular defective trait. For example, if you are going to consider dominant trait here, curly hairs are dominant over straight hairs. Here, if you are going to consider homozygous dominant, that is, both the alleles of that gene are curly hairs. Heterozygous dominant, one allele of that gene is going to be dominant and the other is going to be the recessive. So these are the two possible patterns of the dominance as we are discussing about the dominant inheritance. So here, as per the first point, it is said like dominant expression of the dominant gene is sufficient if only located on one of the autosomes. As we are considering the two homologous, two chromosomes which are which is a homologous pair is going to be present with the two alleles of the same gene. Out of the two alleles, one is sufficient to express. So if there is two also, there is that is one is sufficient to express the gene. That is what the meaning. Next is males and females are equally likely to inherit the defective gene. So here either the males or the females having this dominant gene or contributing this dominant gene to their offsprings are equally likely to inherit this gene. As we are discussing about the genetic disorder, here we are using the defective gene. Here defective gene nothing but a mutated gene or aberrated gene. A parent with a dominant defective gene has a 50% chance to inherit to any sex child have the trait. Now here, as the autosome is irrespectively going to inherit to any of the sex. So if you are going to consider the allosome, it is respectively going to get inherited. That is, XY is for the males, XX is for the females. But here autosomes are not so. They are going to get inherited to or segregated to any of the sex of the individual. So here the parent with a dominant defective gene has a 50% chance to inherit to any sex child have the trait. In the sense here, whichever the parent is taking part in the inheritance, out of these two parents, any of the parent will be equally likely to be inherit the trait. Each affected person usually has an affected parent. Here in the sense, for example, if the offspring is showing the trait, that is the defective trait, if it is normal trait also, it should come from the parents itself. But as we are discussing about the genetic disorders, if the person is suffering from the genetic disorder, as this is being to be a autosomal dominant inheritance kind, the parent is also going to be suffering. In the sense, the dominant gene would be expressed there also. So there is no skip of generation here. So all the generations are going to show the expression of the genetic disorder. So examples are polycystic kidney disease, Marfan syndrome, and neurofibromatosis type 1. Now here, as we have discussed, here you can see affected father. The affected father is having the gene on the chromosome which is lighter in color, and unaffected is this darker in color, and the mother is normal or healthy. They are going to inherit their autosomal genes into their child. Here you can see one is affected son, second is the normal daughter, third is the normal or unaffected son, fourth is the affected daughter. Now here you can see this homologous combination. One chromosome is with the dominant gene affected. Here also the one chromosome with the dominant gene affected, and the other is the normal. Now you can see here there is, in these two children, there is no the defective gene. Here they are not affected with the genetic disorder. Now out of these four offsprings, one is this one and other is this one. These affected two offsprings are going to constitute to 50% of the total probability of the inheritance. So here this is 50% of this total inheritance. Now you are going to say that these affected individuals are going to have an affected parent here. Okay, that is what the meaning, what it is. Affected person usually has a affected parent. 50% chance to inherit to any sex child have the trait. Now if you are going to consider this male, it is going to have this male is going to inherit likely to this and to this offsprings, which are male and female, and the defective gene is coming from the parent, that is the male. If the same gene is going to present in the female, there would be a chance of getting either of these four, any two, either of these four. So here the autosomal dominant is going to express depending upon the pattern, but here whichever the individual is going to get affected, it is going to have a affected parent. And if you are going to see this particular pedigree, in this is the autosomal parent male. It is inherited to a daughter and son, and they are inheriting it to a daughter and son, and this son is inheriting to a daughter. So here you can see first generation, second generation, and the third generation. All these three generations are showing the genetic disorder, so there is no skip of generation. So this is what the meaning. Here if you are going to take this particular diagram in this form, this is also showing the same. So here affected dad and unaffected mother, unaffected child and unaffected child, and the affected child and the affected child. So it is because of the what you call defective gene. Now here you can see the defective gene is with the father, and here it is with the child, and here it is the child. So there is 50% chance of the inheritance. So getting to the next slide, autosomal recessive inheritance. In this we are discussing the hallmarks. The expression of the recessive gene is if only located on both of a homologous autosome pair. Now here, as we are discussing about the recessive inheritance, recessive in the sense, you know that is the other form of the gene compared to the dominant one, which is suppressive in nature. Here if a dominant normal gene is present, that is if you are going to consider the heterozygous form of the inheritance, there the normal gene is going to overtake this recessive defective gene. So recessive defective gene is not going to take part in the expression of the defective nature or the character. Now as per the recessive inheritance expression, the alleles, both the alleles of the gene, of the defective gene is going to be present in the homozygous form. Then only the expression is going to take place. If not present in the homozygous form, the heterozygous form of the individuals called to be as the carriers. So carriers in the sense they are going to carry that defective gene generation by the generation. The expression of that defective gene is going to only when they are going to come across with the recessive gene, which is again the homozygous type. So here there should be 2 copies of the recessive alleles of the defective gene can cause a particular defective trait. Both male and female carriers are equally likely to inherit the defective gene. Here also either the male can inherit or the female can inherit, but to express both the genes has to get inherited into an individual. But for the carriers, that is to carry from one generation to the other generation, the carriers are likely to inherit to the any of the offsprings. Both the parents with a recessive defective gene has a 24% chance to inherit trait to any sex child. Now here again, irrespective of any of the sex, there the parents can inherit the gene. Each affected person usually has an affected ancestor, might be grandparent and above. Here, recessive genetic defect skip a generation and appear in the next. Here you can understand that because there will be a higher chance of the dominant gene to get combined with them to form a heterozygote. So here the chance of the appearance of the recessive gene or recessive defective gene will be very less, up to 25% to the overall probability. So here, because of that, there will be the chance of the skip a generation or skip of generation depending upon the combination of these two defective recessive genes. So here probability of 25%, and there is a probability of expression at the very less percentage. So the examples are cystic fibrosis, sickle cell anemia, Tay Sachs disease. Now here in this thing you can understand in these particular pictures, unaffected carrier father, in the sense one will be the dominant, other will be the recessive gene. This recessive gene is the defective gene. Here the mother, again it is a unaffected carrier, it is the dominant and this is the recessive. Here the probabilities are homozygote, dominant, heterozygote, dominant, heterozygote, dominant, homozygote, recessive. So here this homozygote dominant is unaffected. 1 is the chance out of 4. This is the other. Again you can see, as it is going to have the dominant, they are going to be normal but called to be as the carriers. Here this is not the carrier, this is the carrier of the recessive defective gene. Now here, when you are going to consider this, both the allelic form of the genes are the recessive and homozygous, and it is going to, this individual is going to express the particular genetic trait. The same thing has been put across in the form of the chi-square. Here with the diagrams you can see this individual got affected and all other 3 individuals are going to be the healthy. Out of this, 1 is going to be the normal, this one. These 2 diagonally, these 2 are going to be the carriers. Now we are going to understand the same thing here. These 2 parents are in this pedigree analysis. This is the first generation, second generation, and this is the third generation. Here these 2 parents are the carriers. This is the half shaded structures. This is the male and the female. These half shaded structures mentioned to be as the carriers, and they are going to inherit their genes. Here you can see it is the again a carrier in the sense heterozygous in their nature and inheritance. Here also the carrier female, carrier male, and here it is the affected male. Now, these same second generation individuals are going to inherit. Here again it is the carrier and carrier. First generation has no signs of the affected individual, in the sense the defective gene is present in the recessive allelic form, but the expression has not taken place. In the second generation, you have seen one individual got affected, and in the third generation, there is no individual got affected. The chances of getting affected in any of the generation is very less. Because of that, there will be the chance of the skip in the generation. The chances of getting affected very less, it is because of the recessive gene, which is going to be inherited in the heterozygotic form. So here, as we have discussed here, the point like affected ancestors might be grandparent and above. Here, if you are going to see the third generation, after, if these individuals which are carriers, if they are going to inherit again to the fourth generation, there may be a chance of the birth of the individuals in the all heterozygotic form only if they are going to get married to a person who is healthy in nature like this. So that generation also can get skipped. Like this, there can be the skip of the generation 1 or 2 or 3 depending upon their selection of the individual, how healthy he is for that particular gene, if they are going to take the homozygotic dominant male for that character. So here, if you are going to consider this individual, their parents and their his grandparents can also be the carriers, and his great grandparents can have the defective gene expression. And this is the X-linked dominant inheritance. Here, as we have seen, as we have discussed about the autosomal dominant, the same points are going to hold good here also. That is, one gene, allelic form of the gene is sufficient for the expression out of the two. Men and females are equally likely to inherit the defective gene. That is also the same. A parent with the dominant defective gene has a 50% chance to inherit to any sex child have the trait. That is also the same. Each affected person usually has a affected parent, that is the immediate above generation is going to have the affected parent, as the defective gene is in the dominant allelic form, and genetic defect occurs in every generation. That is what the meaning. Now, except to all those autosomal dominant inheritance pattern hallmarks, the two different points has been given here. The mother alone is the carrier of a defective gene associated with a disease or disorder. She herself will have the disorder of her daughters and sons. 50% will have the disorder. Now here, for example, if you are going to consider this particular diagram, here mother is having the affected gene which is dominant, which is present on the X-linked. It is going to inherit its X chromosome both to the males and the females. Always remember, mother is going to have the two X chromosomes, where during segregation, it is going to segregation of the gamete formation, it is going to segregate it in the form of the monoploid gamete. So haploid or monoploid gamete. So here it is going to get a X chromosome, and other gamete is going to get the other X chromosomes. When they are going to fuse or get fused with the other opposite gamete, either if they are going to fuse with the Y chromosome having the gamete of the male or X chromosome having the gamete of the male. So here these two different fused gametes of XX and XY are going to give rise to the what you call son and the daughter. If those gametes are going to have this defective gene is going to get inherited both son and the daughter from the mother. But if so here because of this there will be the 50% chance of the inheritance of the defective gene on the X chromosome within the all probabilities. You can see if you are going to consider this particular male who is affected and the gene is present on the X chromosome, he is going to inherit his gene to the daughters because the gene is present on the X chromosome and the male X chromosome will be contributed only to the daughters, but the sons are going to get the Y chromosome. So here the out of the overall inheritance probability 50% is going to constitute only to the daughters. So here male is going to inherit its defective X chromosome to the daughters itself, this one and here. The sons are going to remain the healthy. So this is what the pattern of indifference of the inheritance. So here if you are going to consider this pedigree which is having the male affected, he is inheriting it to the females only, daughters only, and the daughter has inherited both to the male and the female, and the son has inherited both to the male has inherited both to the daughters. So here you can see first generation, second generation, third generation and the fourth generation. Here daughter has or the female mother has inherited both to the daughter and the son. Daughter again inherited here to the son. Here the male has inherited to the daughter. So here you can see. So single dominant gene is sufficient for the inheritance. Males and females are equally likely to inherit the gene, and there is the 50% chance of the inheritance of the dominant gene, and there is no skip of generation. You can see there is no skip of generation. The unique 2 points are that mother is going to inherit its defective gene both to the male and the female offsprings, whereas the father is going to inherit only to the female offsprings. That is what the two different unique points. So next coming to the X-linked recessive inheritance. Here the expression of recessive gene is if only located on both of a homologous X chromosome pair. Both affected male and female carrier are likely to inherit the defective recessive gene. Here not equally likely, here only likely. Two copies of the recessive allele of the defective gene can cause a particular defective trait in daughters, but rarely affected has a 25% chance to inherit trait to female child. So there is only 25%. The males are affected 50% of the males by the single X chromosome inherited by the carrier mother. So here there is two different probabilities as it is in the X-linked dominant, but here it is very different compared to the X-linked dominant. So we will discuss this. Each affected person usually has an affected ancestor, might be grandparent and above. Recessive genetic defects skip a generation and appear in the next. So here this is the same point as the autosomal recessive what we have discussed. The examples are Hemophilia A, Duchenne muscular dystrophy, that is DMD syndrome, X-linked agammaglobulinemia. This is a kind of anemia, XLA. Now we will discuss the points. So here one X chromosome, sorry, this is the male, this is the female. Now here when you are going to consider this X-linked recessive inheritance, as the male is having the only the male is having the defective gene which is recessive in its allelic form of expression. It is going to inherit, the male always inherit the X chromosome to the daughters only. Here you can see. So here the daughters are going to get the X chromosome which is affected, or which is going to have the genetic defective gene which is recessive and it has been inherited, whereas the female is having the normal gene. The other chromosome is having the normal gene which is dominant to this particular recessive. So here the individual is going to be the, or the offspring is going to be the healthy. Here also healthy, you can see. So here what will happen, the chance of appearance of the defective gene has to be of having both the individuals the defective X chromosome for the recessive allelic form of inheritance. So if even the female is going to have the same defective gene for the trait expression, then this daughter is going to show the expression of that particular genetic defect. So that is also is going to be up to 25% chance of the expression. But usually what will happen, as the other individual is going to be the normal with the both their allelic form in their dominant expression, then the nature of the expression is going to be the healthy, where these both daughters are going to be the carriers, both the sons are going to be the healthy. So this is what the difference in probability. Here if the mother is the carrier and it is going to inherit the defective gene, if the son is going to get that defective gene and other comprising or the constitution is the Y chromosome, so because of that what will going to happen, the son is going to express the defective character, the daughter is going to be the carrier, and the other two offspring is going to be the normal because their combination is the healthy genes, that is the dominant genes which are not defective. Here in the pedigree you can see the two individuals who are healthy, in the sense they have not been diagnosed for any of the defective gene, but with the pattern of this particular history of the family, it has been understood like this. There would be any of the individual as the proband, with the help of that proband they would have come across this particular individual, the second generation individual, to be as the carrier. Now here if you are going to consider here mother to be as the carrier, so the daughter has got the X chromosome, and further the daughter has inherited to the one daughter, one son and the other daughter, where in the third generation both the females are carrier but the male is the expressing the defective gene. In the fourth generation one male is expressing the genetic disorder and the all other daughters are the carriers, and again in the fifth generation males are expressing the genetic disorder, whereas one daughter is the carrier. So here you can see that there can be the skip in the generation. So here if you are going to consider from here to here, there is the inheritance of the defective gene, but sometimes due to the probability there can be the chance of the expression of the recessive gene in every generation also. But here if you are going to take this same example, I mean to say, if you are going to consider the same example for the hemophilia, and if you are going to consider the different family study compared to this pedigree, as this pedigree is showing a particular family which is inheriting the X-linked in a recessive gene, but for the same hemophilia, if you're going to take the other family, there you will come across definitely the skip in generation. Here why I have selected this pedigree means there can be a chance of appearance of the genetic disorder in every generation, even in the X-linked recessive inheritance. For that sake I have selected this particular pedigree of a particular family. In the same manner for the autosomal recessive also you can observe. This is because if the individuals are going to marry within the genetically related population, during that time there will be the chances of the reappearance of the genetic disorder in many generation, particularly for the recessive kind. For dominant kind, let it be for the autosomal or the sex linked dominant, both there is the chance of the appearance. Let it be you marry within the family, genetically related family people, or out of the genetically related family people. But for the recessive kind, there is the chances of the reoccurrence of the disease if you are going to marry within the genetically related people. So this is what regarding X-linked recessive inheritance. So this completes about the autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive. So here with this we are completing these slides, and we are going to come with the different examples of the genetic disorders related to the autosomal dominant, autosomal recessive, X-linked dominant and X-linked recessive. So and we are going to thank you for tuning the Learner's Big Book, and we request you all to spread our slides or the video logs to your friends and to your other different college or university students if you are going to find our slides as helpful to understand the concept. And also I request to leave the feedback, let it be good or the bad. That is what I need your opinions to make these slides more better and my explanation more better, so that you can understand the things in the better manner. And I hope for today's slides I had been audible, and I request to like our or subscribe to our channel, and also give likes to our the video logs which you have watched. And thank you for tuning to the Big Book.